Okay, that's one. What happens to the foot? Yeah.
It's a blade.
Blade-like foot that penetrates into sediments. What happens to the visceral mass in the shell? Shell is split in two with a hinge on the back that allows it to engulf the entire body of the animal, or surround the entire body of the animal. Okay, what else?
Chitons or yikes. Am I saying that right?
How about we let someone else go since you already got the cephalopods? Polyplacophora. What happens to their foot?
That one, I know the shell becomes 8 segmented.
Shell becomes 8 plates.
Yeah.
Okay, we got the shell. What about the foot?
Has the gills.
The gills, the mantle cavity ends up being along the side of the foot, and the gills are running along the side of the foot. Okay, that's pretty much it. Head is pretty much the same. Okay. Did we talk about any other groups? We didn't talk about the others in detail, but I did draw a couple other groups up there. Monoplacophora, which are actually within the Polyplacophora, even though we had called them their own thing. They're nested within. It's just a Polyplacophora whose 8 plates are fused into one single plate. Okay? What else? Aplacophora, what do they have? No shell, but they have the posterior mantle cavity and the foot and the visceral mass. In fact, some of them do have a shell, but they're little needle-like things along their back. They're pretty kind of cool. Okay, aplacophorans. We had one more that we talked about. Tusk shells. What was the name of that? Scallop? Scapha. Scaphopoda. Okay, what's happened to their parts? Visceral mass is elongated. Mantle cavity runs through the shell and there's an exit on the backside. Okay. What about the foot? I drew a picture, but I didn't put. Foot is like tentacles, arms, and the head is the head. All right. Cool. How are you guys feeling about mollusks? Feeling as good about mollusks as me? I love the mollusks. Okay, so the next group that we're going to talk about, a new phylum here, are the annelida. The annelids. Annelids are the segmented worms, and that's the thing that is the big definition for the big defining structure. They are serial repetitions of coelomic spaces that have little separations between those coelomic spaces called septa. So this, when we're thinking about the themes of organisms, right, we have those themes of what's kind of going on with development and body plans. This is one of the themes. This is segmentation. In annelids, we see segmentation, and some of them we see what's called tagmatization. Segmentation and tagmatization are linked together, along with metamerism. In this case, tagmatization means portions of the body are specialized regionally. So you end up having organs within segments that are specialized for specific things. In the basic annelid plan, every single coelomic space in every single segment has every single part. So in some of the really primitive ones, you see gonads and places where sperm or egg can come out in every single one of the segments. The whole body is nothing but each segment being the same parts, having a nitrogenous waste removal system, having a gonad, having pretty much each of the kinds of things that you might see in a whole body, but an organ in each segment. In more advanced ones, we see those kinds of structures grouped together so that a few segments become the gonadal segments, and a few segments become the nephrostome segments, the ones that are actually going to be doing nitrogenous waste processing. So that's called tagmatization. Each segment has a septum making a space that is disconnected from the neighboring space. which means musculature can work on each one of those spaces. If you squish the circular muscles on one septa, that segment will elongate, but the neighboring segments won't. So you have much more control over the body because you can elongate or contract specific segments separate from neighboring segments, as opposed to like you would if the whole animal were a big balloon animal where you squished one end and the whole other end squishes out the other side. In this case, if you squish one end, it only elongates that segment and not the whole animal. So that gives it the chance for better control of its body and the hydrostatic skeleton. Cephalization, concentration of sensory things along the anterior end. They have nerve cords that run the length of the body. Most organisms other than vertebrates have their nerve cord on the ventral side. Chordates have their nerve cord on the dorsal side, which is kind of weird for most animals. So they actually have the nerve cord running on the ventral side. There are closed circulatory systems with portions of the system that are actively pumping. So we call them hearts, but they don't have valves the way that normal hearts do. They're really just muscular sections of veins or arteries that allow, for pumping of fluids through the closed system. Complete digestive system, so they can continuously feed with regional processing locations along it, and metanephridia, which is a excretory structure that allows for reabsorption of compounds from the coelomic fluid to limit the kinds of things that get lost. So, you can think of this thing as a tube within a tube, an exterior tube that's segmented and an interior tube that's the gut, regionalized so that portions of the gut are for collection and movement, places are for storage and for grinding, and regionalized places for absorption. The metanephridia is this sort of elongated, looping structure. The reason why the metanephridia is so cool, and it's actually something we see in most advanced organisms, is that the metanephridia drains the coelomic fluid from one of the coelomic spaces, and the metanephridia tube runs through the fluids of the second coelomic space. So what you're able to do is have one coelomic space be drained into the tube and then selectively return things back to the coelomic space across the membrane of the metanephridia. So that what actually gets excreted out the side is concentrated waste where the useful compounds have been returned back to the coelomic space of the next coelom, of the next segment. Yeah.
Wait, so it goes from...
So think there's a septa, a hole on the septa, so the fluid from one coelomic space drains into that hole, but the metanephridia loops within the next coelomic space. So what's in this coelomic space is regulated, or what's going to be released is regulated through that tube, returning things that are useful back into the coelomic space of the next segment before it drains out of a hole on the side. So if you've ever picked up an earthworm and it's all slimy, that's actually coelomic fluid, or it's urine, seeping out of holes along the side of it. So when you pick a slimy worm, it's covered in worms.
Each individual segment
has
responsible for its own.
each individual segment is responsible for dealing with the waste of the next anterior segment. So the segment before drains into the tubule that runs through the space of the next segment. And then that segment has another one that runs through the next space of the following segment. So it ends up being a really good reabsorption system. It ends up being a way that, if you think about what was happening with the flame cells and some of the other organisms that we've seen, or even in the mollusks, there's just one pair of nephridia. There's only one chance to make a regulation of what gets reabsorbed back in the animal. In annelids, it's every segment. every segment has the potential to reabsorb important compounds that you wouldn't want to lose. In the circulatory system, they've got these anterior vessels that are enlarged and muscular and serve as hearts. There's a blood vessel that runs one along the dorsal side, one along the ventral side, and then connections that run around the gut for each segment. So each segment can be supplied by blood that is carrying both oxygen and food. They don't have specialized gills, although some of the marine systems do, but they can diffuse their gases directly across the body wall. But because, if you remember, the distance of diffusion can't get into the interior of the organism, but just below the surface runs those closed circulatory systems. So as the oxygen or gases diffuse into the body across the body wall, they come in contact with this blood that can carry that oxygen. Likewise, on the interior space, as food items are digested and moved, and those compounds dissolve across the gut wall, they come in contact with the blood and are able to be carried around to the interior cells that need it. So the closed circulatory system increases the ability to supply the interior cells with the gases and nutrition that they need for functions. In the most simple setup here, there's a nerve cord that runs ventrally, where each segment has its own little segmental ganglion. So each individual segment can be regulated separately from every other segment. They also have a brain, and that brain loops around the esophagus. Remember, we were talking about how most brains are actually circular, running around the esophagus. In most of these organisms, these guys have it too. There are these cerebral ganglion that act as the brain, but they're actually part of the loop that's around that thing. In most cases, they're sexual reproducers, although there are a few asexual versions in marine systems. They're generally dioecious. What does that mean? There's another word for that, gonochoric as well. Separate sexes. They're often hermaphroditic, especially in earthworms. For the most part, external fertilization is what takes place in things like polychaetes, so we'll talk about that in a bit. They very often spew their water into the water column, making sperm. And the general larval form is a trochophore, which is that little tufted, top-like looking larvae that also is shared with the mollusks. In fact, when you look at molluscan development and annelid development, you can't really tell apart which is trochophore for one or the other, except for this set of 8 cells that form a little cross on the top of the tuft at about the 164 cell stage, and that cross has a distinct look if it's a mollusk or if it's an annelid. So you can't really tell them apart until then. The way they move is by altering contractions in individual segments. So by contracting the circular muscles around a segment, that segment elongates. By contracting the longitudinal muscles, the segment contracts. So if they want to move forward, they'll contract the circular muscles, and that pushes that segment forward. And then they flare, or they flare that segment by contracting the longitudinal muscles, and that sets like an anchor right at the beginning. And then that contraction goes as a wave from segment to segment to segment, pulling along individual segments and ultimately pulling along the entire organism. So if you look at this sort of movement this way, you contract the back, this forward part pushes forward, and as the movement of contraction moves towards the back of the animal moves forward in a direction opposite of the wave of contraction. It's simply alternating contractions of the longitudinal and circular muscles. Every segment has a set of chaetae, or sometimes they'll be called setae. It's just a chitinous or keratinized extension that sticks out the side of the body wall. Those things are often used for grasping and holding in segments. So as as you flare out your segment, those chetae hold into the side of the soil and keep that segment from moving when the contraction wave heads down the animal. We have three major groups, three major classes. Most of the annelids are actually marine that you probably have never seen or experienced, and those are called polychaetes. Chaete means the chaete, right? So poly means lots of chaete. And they have these little appendages sticking off the side that are called parapodia. This is the oligochaetes. The oligochaeta are the group you're probably most familiar with when you think of a worm. This is the worm you think of, right? These are earthworms. They're both freshwater and marine. And this guy up here is the Hirudinea, or the leeches. Did you guys know leeches and earthworms are really closely related? No? Earthworms and leeches are very closely related. Okay, so let's talk about polychaetes. Polychaetes are really pretty diverse. 5,000 named species, but it appears as though these things are riddled with cryptic species. One of the first groups of people who recognized cryptic species was somebody on my committee for PhD. Her name was Judith Grassley. She worked on a group of polychaetes that are called, I can't remember the name. It's not important. Anyway, she was working on this group of polychaetes that were considered cosmopolitan. You basically find them everywhere globally. You go and look in marine sediments, you find them. Capitella, that's the name of it. And so she was interested in their genetics, and she collected Capitella capita, one species, they all look the same, from a bunch of places in Massachusetts Bay. So one bay, one location, one species. 6 different spots in the bay. In every single one of those spots, she found a different species. Can't tell them apart because they look identical. They're basically living in the same kinds of environments, so nobody had thought that they were something different. But when you look at their genetics, they're different species. And so our expectation for at least Capitella is that what had been one global cosmopolitan species is probably more on the order of 200 different species. Okay? So, if one of the most common is actually 200 species, how many more within those 5,000 are actually separate species? And chances are it's a lot more than anyone realizes. One of her students, who was a friend of mine in graduate school, she was working on a different species that lives in the sandy coastal plains off the coast of New Jersey. And she found that in these places where you get little ridges of sand, no more than an inch high, little ridge that's an inch higher than the trough, there is a partitioning of that habitat from what had always been considered one species of polychaete. There's actually one of the polychaetes lives at the top of the ridge and another species lives at the bottom of the ridge, but you can't tell them apart when you look at them. So chances are there are a lot more than 5,000 species of these things. Almost all of them are marine. They've got these parapodia sticking out the side that looks like, remember what para means? Near, podia are feet. They're near feet. They're not real feet. They're near feet. Parapodia sticking out the side with little chaete hairs. Their potential ecologies fill pretty much everything you can think of for marine systems. There are ones that crawl around in the sediments and eat the sediments. Those are called deposit feeders. That's a lot like what you would think an earthworm is. There are deposit feeders that live in a burrow and stick their little parapodia out and pick up sand and eat it. There are ones that swim around and are pelagic predators. There are ones that are raptorial predators that have these huge jaws and grab. I even showed a picture or a video of a bobbit worm in lab. Bobbit worms eat fish. So the fish swims by and the worm comes out and grabs the thing and pulls it into its burrow and eats it. Pretty much every single kind of thing, filter feeders, filter feeders who create a flow and make a mucus bag that gets filled with all those things and then eats the bag. Everything you can think of ecologically that a marine organism might do to make a living, there's a polychaete that does it. For the most cases, they're dioecious. Most don't have permanent gonads. They only develop gonads right before mating. And a lot of them do this really bizarre thing that's called epitope, where they don't have gonads, but then they grow gonads in a certain in the posterior segments of their body, and then those posterior segments rip off and swim away from the worm. No head, no brain, just a bunch of gonads in a huge set of segments that swim away and then spew those gametes into the water column once they're up into the water column. So I described this in lab the other day as if your ovaries or testes just ripped out of your body and walked away and went and had their own little mating party somewhere else, leaving you behind. Epitope. A lot of the marine ones do that. They have free living trophical life. Polychaetes are really cool, but people don't really know much about it. General public doesn't really know much about polychaetes. The next class are the ones that you probably do know a lot about, oligochaetes, or earthworms. I have about 40 to 50,000 earthworms in my basement. If anybody would like some earthworms, I can lend you some, or you can have them. I've got a whole composting system in my basement. I can handle about 5 to 10 pounds of food waste per day with my wormery. Well, it's not that big, but it's filled with mushroom logs and worms and my my washing machine. What's kind of cool about earthworms is that much of the earthworms you're exposed to or that you know of, are actually not native to the United States. If you think about what happens every time there's a glaciation, the glaciers push down across the land, and no earthworms are going to live there. So where we are right now, we're under glaciers 10,000 years ago. And so there were no earthworms here. So all of the earthworms that are here now have migrated either from further south, but they've actually mostly been outcompeted with non-natives that were introduced from Europe. So most of the worm species that we see around here are actually European imports, brought with movements of plants and animals from around the world. The driftless region, though, which is this little portion around the Wyoming or around the Wisconsin River, driftless region in Wisconsin was never covered. That's why they call it the driftless region. It's never covered with a glacier, and it actually has one of the few North American natives that's still established, sitting in this little patch around the Wisconsin River. That's like an interesting trivia tidbit, really. Most of them are scavengers, deposit feeders. They ingest sediments and digest out of those sediments things like rotted vegetable matter. So many of them work their way through the sediments and then ingest those sediments as they're working along the way. There are a lot that are actually aquatic, freshwater aquatic, that live in the water. So you know how when it rains and they all come up? It's not because they're drowning in their burrows. They can actually breathe just fine. Because they gas exchanges across their skin. We are starting to think that it's a dispersal mechanism. That when it's raining and the surface of the ground is wet, they're not in danger of desiccating. And so what we're seeing is locations of high population. Worms are dispersing away from those locations. They're coming to the surface and crawling away so that they can try and find a place that are not, where they're not going to be under those density-dependent effects that are probably going on in those individual locations. The way these guys mate is really pretty cool. They are hermaphrodites. Each individual has both male and female parts. And when they mate, they swap sperm between both individuals. But their fertilization is external. So this little thing you see, the thickened sort of band on the side of the worm is what's called the clitellum. And the clitellum is a gland, it's a gland that secretes a cuticular layer that is able to move over the animal. So what happens is when two worms are mating, they find each other and line up in such a way that their seminal vesicle and seminal receptacles line up. And each individual worm puts sperm into the neighboring worms, into their partner worm's seminal receptacle. When the clitellum secretes the cocoon, or secretes the cuticle layer, it begins to break off the clitellum and slide towards the head. And as it's sliding towards the head, it passes over the oviduct. And the oviduct places eggs into the cocoon. So you have unfertilized eggs, now exterior to the body of the animal, in the cocoon. The cocoon keeps sliding anteriorly towards the head and heads over the seminal receptacle where all the sperm has been stored. And then the sperm gets put into the cocoon that's filled with eggs and fertilizes the eggs in there. So you've actually fertilized the eggs exteriorly to the worm inside the cocoon, and then the cocoon keeps on sliding anteriorly until it pops off the head of the worm, and inside the cocoon are the fertilized eggs that are going to develop into the worms. So little baby worms go through their first sets of growth inside the cocoon, and then when they break out of the cocoon, you get little baby worms that crawl out. So if you ever have a wormery, like I do, you can go in there and find all the cocoons and pick them out, and then put them in a new bin and start your new worm bin with a bunch of cocoons from a previous worm bin. That's kind of cool. What do I want to tell you about? We talked about the Metanephridia. Look, here's their mating pair. Look how they line up, and each of the things go in. I just talked about all that stuff. Okay, so the last class of annelid are the leeches, or the Hyrudnia. There's only about 500 species of these things that are named, and almost all of them are freshwater, although there are a few that live on very moist land and in very moist sediments. They are ectoparasites. They attach to and suck the fluids out of hosts. But they do it in such a way that is that's kind of cool. So first of all, they've got these chitinous jaws that when you see them up close, they look like little blades that run at a 90-degree angle to each other. And when they bite, they cross over each other like this. So you can imagine like having a couple of razor blades that go right? They basically just chop open the blood vessels that they're around. Has anybody ever been bit by a leech? You don't even know it when it happens. You like get bit, and at some point you look down and you're like, oh, there's a leech on me. Because they have a analgesic. They put a painkiller on before they bite you, so you don't even know that you get bit. And they also have a protein that mucks up the blood clotting cascade. It blocks thrombin from coming together and forming the clot. So you don't know you got bit, and you keep on bleeding afterwards. And oftentimes, when a leech bites you, it's the size of a hair. You can't even tell that it's there. And then after it's been sucking your blood for a couple of days, you look down and there's this big floppy leech on you, and you're like, where the hell did that come from? It had been on you for a long time. What's really cool about these guys is that most leeches can survive several years without feeding. They don't need to feed very often. And they're also incredibly effective at turning that food directly into eggs. Something on the order of like 95% of the mass of a blood meal is translated into egg mass when they have offspring. And they're that efficient at turning what they eat into new offspring. They are just like oligochaetes, or they're just like the oligochaetes in that they have reciprocal insemination and a clitellum that forms a cocoon for the young. And in fact, we group these two together, the oligochaetes and the Hyrudnia, into a group called the Clytellata, because they have the clitellum. I actually have a friend who grows these industrially. He grows leeches for medicinal purposes. He sells the leeches to a pharmaceutical company that grinds them up and extracts the hyrudin, because hyrudin is really good if you're doing something like doing an operation where you don't want the blood to clot while you're actually in there working. So you can expose it to hyrudin while you're working, and that won't clot. You can keep on working on the thing. Yeah.
Is hyrudin the painkiller or is it the blood?
No, hyrudin's the anticoagulant. The analgesic is something different. All right. Do they have any? So that's the annelids. Those three groups, segmented rows. Boy, that's weird. I literally spend a week on annelids in invertebrate zoology. To do it in 20 minutes is kind of freaking me out. Okay. In this same group, the Lophotrochozoa, there is a group of phyla that are more defined by their feeding appendage than by having a trophophore larvae. So the larvae of these guys sometimes look like trophophores, but sometimes don't. So we're going to look at this group together that we call the Lophophorids. There's multiple species and multiple phyla in the Lophophorids. But the thing that they all share is a lophophore. When I was in college, the Lophophorids were thought to be more closely related to the deuterostomes than the protostomes. They have a lot of things about their development that appear deuterostome-like. For example, deuterostomes, who we're going to talk about in a few days, have a three-part coelom. Most of the Lophophorids have this three-part coelom. They have determinate development, which was one of the things that we said was a deuterostome trait. So we had always thought they were more closely related to deuterostomes until the molecular evidence started piling up that argued that they were more closely related to protostomes. And so now we include them with the other protostomes and the Lophotrochozoa, even though a lot of their developmental traits would suggest otherwise. So what they have is an appendage called the lophophore, and this is a tentacled ridge around the mouth, you're looking at it inside of this thing, that acts as both a functional gas exchange, functions for gas exchange and for collecting food. What did we see that looks a lot like this thing already? It actually does something very similar. Looks a hell of a lot like a bivalve, doesn't it? And we already know that bivalve gills act as both the gas exchange and as the feeding appendage, collecting food and moving it to the mouth. This group, the lophophores, have a secondary, it's a convergence. It's a convergence of body form and plant that are from different evolutionary origins. So there are three major phyla that we're going to talk about. There's more than that, but we're only going to talk about three. The first ones are the Phoronida, the Phoronids. Phoronids, there's not very many of them alive now, but there's a pretty good fossil record of them. Phoronids live in a tube, and they've got this deep U-shaped gut. So you'll notice that they've got a mouth up here, their gut runs down into the body, and then back up, and the anus is to the mouth. Most of these guys are small, and they live in a chitinous tube. Their lophophore sticks up out of the tube and acts as the gas exchange and feeding appendage. This group, a new phylum, looks very similar, but actually has a different evolutionary origin. This is the Ectoprocta. In ectoprocs, they have lophophore, and most of them live as colonial groups. So what's the convergence? This has a convergence to another group. What does this look like to you? Bunch of tentacles out feeding an individual. What's that? Looks a lot like hydrozoa or anthozoa. Looks a lot like colonial cnidarians. And in fact, their feeding style and their lifestyle is the same, but they're not polyps. Each individual one of these is a zooid, it's called. So this container that's in here is a zoosium, which is usually chitinous. There's two major groups here. There's the ectoprocs and the entoprocs. So what does proc mean? What do you call let me use a medical term. A proctologist. What do they study? Yeah, colon, the rectum, anus, right. Proctologist. What's that?
So it's intestines on the outside?
So, if you look at the lophophore, right, what you're looking at is the location of the mouth compared to the anus in the lophophore. So, in the ectoproct, there's an ectoproct and an entoproct. Where would you expect the anus on the ectoproct? Outside of the lophophore. In the entoproct, the anus is inside the lophophore. So imagine, right, in both cases, your anus is right next to your mouth. So, how do you deal with life if your anus is right next to your mouth? Oh, that was good, yeah. Credit on that one. You have to think about how water's going to flow through the lophophore. If your mouth is inside, you want to make sure that the water hits the lophophore first before it goes over your anus. So your mouth inside next to your anus flow is going to come from the outside across the lophophore and up and out. With your anus on the outside, you want to make sure that the water hits your lophophore before it hits your anus. So it comes down across the lophophore and out, going across the anus second. So it's actually kind of cool in that these two groups are separate phyla who have come upon essentially the same plan, and the only way that they're able to deal with the difference of having their anus inside or outside of their lophophore is the either flow going down from above and out to the sides or up from below and out to the top to make sure that you don't foul your lophophore with your own wastes. So these guys look like pharonids because they got this little U-shaped gut and a lophophore, but they're actually a separate phyla, not related. Close, not in the same group. This last group we're going to talk about are the Brachiopoda. The brachiopods are a immense fossil group, but only have about 300 that are alive now. But there's over 10,000 fossil specimens of brachiopods. Most of the things that you find that are fossils where people think are bivalves are actually brachiopods. And the easiest way to tell whether something is a bivalve or not is, does it have this pedicel coming out the back of the bivalve shell? If it does, it's not a mollusk. So you'll notice there's a pedicel here coming out of the back of this shell. Looks just like a mollusk shell, but it's not. The pedicel lets you know that it's not. This looks like a bivalve, but it's not. The pedicel lets you know it's not. When you find these things, you often find them in mass aggregations. So of these, so like this guy, the Lingus here, they're found with tens of thousands of individuals in a square meter, which is a lot like what you find with bissally attached, those are mollusks that are attached to hard substrates. So also a convergence on a similar lifestyle, similar body form, even though they're not closely related. All right, so what have we dealt with so far? We have dealt with, this is our older group, and this is the new group, right? So I'm showing you both trees from before we used molecular stuff and after we used molecular stuff. Did I just dab?
You gotta tuck your head.
My kids are so into it. I'm like, what are you doing?
Like 2 years ago, that's fine.
They're 10 and 8. It takes a while to trickle down to elementary school. So what have we dealt with? We have dealt with the lophophorates here, which are now grouped within the Lophotrochozoa. We've dealt with the rotifers and the pseudocoelomates, who are over here now, are all part. Basically, we've dealt with this whole group right here, the Lophotrochozoans. Notice most of these phyla we actually haven't looked at. Sipumpidans, Ecurians, we didn't talk about any of those guys. Pongophorans, didn't talk about any of them, did we? All right, now let's talk about this group, the ecdysozoans, which are the arthropods and their relatives. These are not up. I'm only going to start like the first several slides, and we're just going to talk about the first couple of groups and the basic lines of them, and then the slides will go backwards. What's that?
You were like, yeah, we're only gonna cover the first several slides.
3 or 4?
Depending on how much you talk on a slide, it's like 2 pages of notes.
Yeah.
Yeah.
What are you saying?
It could be a lot. You just made it sound like a little.
So are you saying no when you start?
Is that a trick question?
Well, I mean, I did just push the exam back, right?
Oh, then you do you. You've done enough for us today.
Because I got like, there's a lot of arthropods. Yeah, okay, why don't we just call it? Yeah, let's call it. I'll start arthropods Monday next week.
Wait, are you serious?
Yeah.
We're all totally blown.
I'm serious. What is that, 15 minutes early? 17 minutes early? This will never happen again.
Hey, you know what? Let's have a quiz today.
We were?
Yeah.
Guess what, we're gonna have a next Friday.
They've always gone Friday.
And that means we won't miss a quiz before,
Yeah, we'll have school.
just because we don't have school doesn't mean that
Quiz changes.
That's it, it's in the, did I have it scheduled for today?
Yeah.
Yeah, I should
Yeah,
probably
you
do
do.
that one.
Oh, I was told.